Hello! Welcome to Microbiology Journal Club. I'm your host Fazalam, and this is my the other host, Danny Chan.
Hi.
And
Yeah, this is a series that we've been doing for a while where we talk about papers. We want to open up that journal review process to the general public. So it's our on week this week where we dive deeply into a paper, but on other weeks we sort of review some of the headlines and try to choose a paper to have this deep dive into.
yeah, and this week we'll be deep diving into Regeneron,
Mm.
this brand new amazing miracle wonder drug. The
Only for Transformers.
yeah,
Only for
so
Transformers.
yes, yeah. So yeah, this is a drug that has been hitting the headlines recently as a potential treatment for COVID, and
Because of a very famous patient.
yeah, a very. I mean, is this
Infamous patient.
infamous patient? Is this drug good enough to save? Are you bad enough to save the president's life? This. Yeah. So can this is it going to be this drug that saves us all? Perhaps. Maybe. I don't know.
But it was interesting for us to choose because we haven't talked about we haven't talked too much about monoclonals previously. I think maybe in our mouse model discussion we had a discussion about monoclonal antibodies. But yeah, we haven't really
Yeah.
looked at a paper where the focus is sort of developing this type of therapy.
Exactly. This was something that was like kind of on our to-do list, and the news story basically prompted us to finally take the leap and go into this paper that describes how this antibody, which is this antibody cocktail called REGN-CoV-2, which is being trialed in various patients in clinical trials and in the president. So, I mean, I wouldn't usually take that chance with an experimental drug on someone really important, but I mean, why not? But yeah, this paper actually goes into how this drug was effectively like discovered and devised, and what were the decisions on why use an antibody cocktail? Why choose these antibodies? Where did these antibodies even come from?
Right, right. Yeah, very much like a development of the therapy in this. But not really anything about the efficacy.
Yeah, yeah. This is very much an early stage paper. The efficacy will come out with phase like 1 or 2 clinical trials. So I mean, you can't really predict. We can't really say how much effective it is based off of just one patient. But
Yeah.
especially since that one patient we know about has got a lot of other treatments as well.
Right. And a lot of like sort of constant monitoring, like a sort of like
Yeah.
a level of healthcare that is probably above what other people are going to be getting.
And of course, this is a disease where some people do just get better from sometimes. So
Very true.
there are many things that could be happening. But
Yeah. Oh, and I guess the other important thing is, like we have no idea how this therapy would behave in like a wider population,
Yeah.
right? Because like knowing stories about just one person, that's the data point of one, and like your genetics don't match up, your health conditions don't match up to that person, and so who knows, right? If you took it, there could be all these effects, and it's just like without wide testing, you have no idea
yeah,
what that's going to
we
be.
have no idea what this will be, how this drug will work in a healthy, mentally stable young adult. So, I mean, we have no idea. So that's something that we'll have to look into for the future.
Yeah, so let's see. The first thing that we have here, I just wanted to highlight to people that they say studies in humanized mice, right? So, like, what does that sort of mean? Or like, why is that even important to highlight? And that's because antibodies, we all know them as Y-shaped proteins, but they actually have some differences between species. Here are like 3D images of antibodies. And you can see like there's just some shape differences that come out. They effectively work the same way. On the two Y arms,
Yep.
they stick to things, and then on the Y stem, that is something that your body's cells recognize. And so that could be effective because it means that maybe your body's phagocytic cells could eat that object. But antibodies could also work in other ways. We talk about neutralizing antibodies a lot. We have been talking about neutralizing antibodies a lot. That's just like, it gums up the works, right? If you have something that sticks between two surfaces that should be matching together, the antibody can block that. So they use these humanized mice Oh, I see. The next image actually, there's all these different parts of the antibody that we're looking at, and with inside of a normal like antibody response, your body's making all sorts of things. We talked about a little bit that in the germinal centers paper. And those antibodies, you just get a whole bunch. It's based on evolution. But this term monoclonal means that they just choose one.
Yeah.
Yeah.
So in the development of antibodies, it's weird because they do have this like RNG moment of like how they're developed where. So
yes,
absolutely. You can see on this diagram there's like different parts of the antibody are labeled like VH and VL, and these are the variant sections that and there are the ways the gene is like structured is like almost like Lego pieces that are related to each other, where you've got like different VHs. I think there are like about like 10 different families of them, and then there are little variants within that. And then basically during the process of antibody production, they kind of like rolls the dice and like puts them together. And then through a series of like focus evolution, they figure the body itself
choose the best ones.
Yeah, they choose the best ones.
Yeah. Yeah, I like that idea that it's Lego. And like actually really interestingly, you can also think that and people have variants of those Lego bricks inside of them. Right? So like that gives rise to I mean like and those areas they're the Lego pieces that are being slid together, they're flexible regions. And so like it's kind of it's weird. It's like Legoing together all these like sort of different sticky pieces, and hopefully in that mix of different sticky things there'll be the sticky thing that you want.
Yes.
The thing that binds to the antigen of choice. Yeah.
So I guess.
What is there anything else we want to highlight here? Oh yeah, maybe the next one actually zooms in on that variable region.
It does zoom in a bit more. So.
Yeah, you can see it's actually there's a really specific. So there's these two arms, the heavy and the light domain. That's just. I think it's just based on how big they are. Yeah. The heavy one is like a bigger chunk of the antibody. The light one is the smaller chunk. But it's right at the tips of these variable domains. There are these flexible loops, complementarity determining regions. And complementarity, that's complementary to the antigen, right? Yes. That's what we're saying here. And so, those are flexible amino acid chains that are the determinants. The actual physical spots that of the antibody that are interfacing with the antigen.
I guess,
and that's the setup, right? Because they talk about these. Yeah, they talk about these spots in the antibodies. So like that's why we just want to give you that background so that when they're talking about these antibodies, you know what's going on.
Yeah, we are going to revisit this. But, I mean, the big question is how do we make these antibodies? And the only way we can really make them is in live animals or in people, because you can't really do this in petri dish. So they got these mice called the velocimmune mice, like, which was...
TM, or like
trademark, it's throughout the paper. And unfortunately, they're not half mice, half velociraptors, as I first thought, but they are, in fact, humanized. So they express human-like antibodies, I believe.
It's that whole system, like all those. So every animal has their own. Every animal that has an adaptive immune system. So vertebrates, all vertebrates have their own system of like Lego bricks that come together, right? And then they have, of course, their own constant regions and stuff. So in this mouse, it's a very extensive genetic engineering project that they took on with these mice. 6 megabases. Of mouse DNA were replaced with the human counterparts.
That's 6 million bases that they spliced out and they put in. And then they took these mice and they gave them a vaccine where they gave them a DNA vaccine followed by versions of the spike protein binding, so RBD. So it's like vaccine, but of course they spend maybe 2 lines focusing on this, where we've looked at papers which spend all the time focusing on this, but they do it in a group of mice, and then they get and they checked the antibody titers and found that they had antibody titers to
this is figure S1, right?
Figure S1. Yep, figure S1. They've got antibody titers to the RBD, which shows that they make antibody against it, which is good, and this antibody will be very human looking, so that means it might be useful for human studies, which would be also good.
Yeah, we've talked about this before. The way to measure antibodies, they're just doing ELISAs here.
Yeah.
You get a bit of the receptor binding domain because that's what we think will make productive neutralizing antibodies. And see what sticks.
Yeah. And they actually do, unlike the Russian vaccine, they actually use an odometer to measure it. So
we're seeing that variable,
Yeah.
variable nature, not this discrete
And so
dilution factor version.
Yeah. So this is where they're getting their antibodies. They vaccinated 32 mice and they pulled out a bunch of stuff from them, and they also took 3 people who were symptomatic of SARS-CoV-2. But then afterwards, after they'd recovered, they took blood samples, and so So
And I think there could be we're saying that these mice are humanized in so much as the antibodies that they make are human looking.
Yes.
Right. But that doesn't mean that their sensing systems are human looking. Right. Like there's differences in our toll-like receptors and, intracellular sensing, and like all of that can make different antibodies appear. Right. I think the germinal centers are probably like a little bit different in mice, right, than in humans. And so we don't know. There's different ways of getting the antibody, the neutralizing antibody.
And the thing that is very different about these, because we have talked about humanized mice before, but these mice don't have the human ACE2 protein, so they wouldn't get that same infection. So they'd get a much more milder symptoms if they did get any from the coronavirus, but they would So we're still banking on them having a good immune response. And with the SARS and with humans, they used 3 donors. So 3 isn't a big number, and the immune response can be quite diverse. So we don't necessarily know whether there's any, if there is a unified best response, whether we'd be able to pick it out from just these 3 people. So yeah, from the mice they took out the spleens, and from the humans they took blood samples, and from those they extracted B cells. So, and they put these. So we've talked about flow cytometry extensively in one paper about how in the journal paper where like they separate out different cells based on what they bind
the different populations of the cells.
Yeah. So effectively that's what they do here. They separate all the different populations of mouse B cells, and then from them they take out cDNA. So that's basically they take RNA and they convert it into cDNA, and they use some specific primers. So that's the kind of thing that'll read the DNA and take out copies. So they pulled out the variable regions, the antibody basic genes from them.
Yeah. So inside of these B cells, I mean, definitely in the humans and presumably in the mice, right? That the B cells have been activated, right, by seeing other parts of the immune system have identified the virus or the vaccine in the mouse's case, and they've been like, send the alarm, we need to make B cells.
Yeah.
And those B cells go through that process of some of them are random. They all have the different random Lego bricks assortments, right, of making different antibodies. The certain ones are selected. They pool in the blood or the spleen. We've got those out now, and then we also have their sequences. So it's kind of like a reverse engineering style, right? It happened in the body, and now we've extracted the information of what the body did in response.
Exactly.
What did the body choose as the best antibodies?
Yeah, we're basically cribbing off the body's notes, and that's what the CDA is. We're looking over the body shoulders, taking it, and then putting those into Chinese hamster ovary cells so that they can produce the antibody, and then we can subject that to further testing. And that's how they came up with the antibody. And this paper is how because they came up with a bunch of antibody candidates based on this. I think over 1,000 that they filtered down to 200. Well, I mean, Figure 1 sort of gives you an idea of that, right? Because I think the numbers are how many antibodies fell into those categories. Yeah.
So it's it's a little bit different because it's this is heavy chain versus light chain identities. There's oh my goodness. So I'm not sure if we said this before, but there are isotypes of antibodies like IgG and IgM. Those are there's all these different types of antibodies. Then even within those types of antibodies, there are types of the chains that make them up.
Yeah.
And so that's what we're seeing in Figure 1.
Effectively, yeah. So this is them characterizing all the antibodies that they managed to filter down. So this is already a filtered group from about 200. And then they
Oh, I see. Yeah, this is the filtered from that.
So, and this is them characterizing basically what Lego bricks make up each section of antibody and how they pair up. So like
not the exact.
So the VH families and light chain and heavy chain, and so it might be a bit different. So I've like put some lines in to make it a bit more easier to pick out, because there are some ones that they like to look at, which is so the IG8V353, which is quite an important one. So in previous studies, that's been a subtype that's been found to be quite important, or that shows up a lot in people who are infected with SARS-CoV-2. So the idea is that little Lego brick is much more able to develop into something that can bind to the spike protein.
Like something in that hard-coded region is like the spike protein.
Yeah.
Because there's also, I believe there's another step as well. Like there's also more mutations that accumulate inside of these. It's not just the Lego bricks coming together. There's also more mutations. So but it has the right raw starting point, we'll say, right? That genetic locus has the right starting point.
Exactly. And they found that the mice tend to have quite a lot of antibodies in that sort of region that were focused towards SARS. Humans less so. Humans were more focused on. So I've highlighted the HV353 in yellow, and then the humans, the three people obviously, so much lower, and they tended to go with HV366. But then again, the three again, we can't really tell much from numbers, but this like kind of finding from mice does support other research that's out there.
Yeah, I guess I mean, this is the big term I think that gets thrown around. They use it in the paper. They call the T-cell repertoire, right? It's like, or T-cell B, whatever, antibody repertoire. Antibody repertoire. It's like, it's that term that tells you like what are all the possible things that are made, right? Like from the body, because like not everything is gonna work well in selection, and you see it here, right? Like there are certain hotspots, of these genetic loci. It's kind of interesting. I don't think that this information is really, to me, I don't get any more or less information seeing this of like how this cocktail works. I think it's, as you said, it's mostly like how does it line up with other observations?
Yeah.
Kind of like the basic science slant of this, right? Like even though we're going to end up talking about a therapeutic in this paper, actually there's a bit of investigation here just to know like, what sort of things go into a productive immune response against this pathogen.
Yeah. I mean, the thing is like, at this point there is like some disconnect because the antibodies we end up with at the end of this, it's not really clear which pool they came from and whether they came from mice or human. So
true.
So that'll be something to look into. So, but I think this is just interesting for scientific merits, but not necessarily for the treatment side.
Yeah, exactly.
So the next one we're looking at is this focuses in a bit more on the antibody.
Which again is like not super therapeutically relevant, but perhaps for people who study antibody repertoire could be sort of useful to know.
Yeah.
So that IgH and IgK, IgL, these are those isotype.
Yeah.
This is the same information in some ways, but now we're looking at the frequency that they each occur in the antibodies that they found.
I think the main story they wanted to say here is that the human and the mouse antibody structures and isotypes are very similar, and therefore kind of putting forward the argument that their vacuum immune mice would be a good basis for developing new antibodies. So
I definitely, well, the whole fact that those mice are TM'd, like that they're a product. I think that that's a really good way of interpreting sort of this beginning part of the paper, right? Is trying to show that this product that they've made could be used instead of needing human samples.
Yeah.
Right.
And I can see the argument there because you can 36 versus 3. You get this much bigger diversity, and you can get a lot more kind of resolution on specific isotopes that are very potent against.
Well, I mean, my thought is that they're trying to position the model as something that can be used for stuff that the infection's not even rampant, right? You don't have to have a sick person who's willing to donate their stuff. You can just infect some mice with the stuff that you want to make antibodies towards. And maybe also importantly is that monoclonal antibodies aren't just used actually, there's very few cases where monoclonal antibodies are used in an infection. Like a lot of the use of monoclonal antibodies we see is against like immune targets, immune checkpoints and stuff like that. So it's like they're immunomodulatory drugs, not like anti-infectives. So, maybe one of the things that they're trying to get at here is like, whatever you want to inject in our mice, you're going to get antibodies just like the humans would make.
Yeah.
Yeah.
And with D, we've got another comparison on the complementarity.
Yeah. So is this the length? This is the length of them.
Complementarity.
Yeah. I guess knowing the difference between human, it's the same legend as the last graph, that the dark lines are the mouse derived and the light lines are the human derived.
Yeah.
So I guess maybe that's the comparison they're trying to bring out here. That they're similar in that way.
So essentially, the big peaks are supposed to be in the same place, but since they have so many more mice, then the black peaks are going to be higher. But this is more like selling their model.
Selling the model. It's like very abstract from the sense, because it's also like, what does it mean that this specific length is the length that's most common? Like that has almost nothing to, right? That's just because of this antigen, actually, right? Different antigens may have different things, or even I think immune systems probably have a preference. Like there's like a sweet spot probably that like these CDR's lengths are. Yeah,
it would be used to have like a comparison with something that doesn't bind to COVID, so that isn't just like a general, this is the length that all CDRs are usually, or it's within that kind. Just see how far it is away from the average, because then that would be quite
I feel like there's like a pool of primary literature that I'm not super familiar with, and again, it's this repertoire. It's about like antibody repertoire literature. Like, what does it say about, right, like the characteristics of these repertoires and how that relates to immune system function? Like, I don't know those answers, and I didn't dig into it for this paper, but that's what this data is used for, right? Like that's what people would be looking at.
I mean, I would argue that there is some like kind of relevance to the length, because in terms of like the actual flexibility and some structural aspects of it, like I think that there is something there, but they don't really.
Well, I mean, you could say that the longer it gets, it's less effective, right? Because it's like there's so much flexibility that it's not specific to bind something like that. Yeah. That's a biological thing that you can see in this, but like how that relates to the specific antigen, I'm not really sure.
I'm not sure either, but I mean, again, that was the point of this, was to say, look, humans and mice, they look very similar. Can't we all just get along?
Yeah. Don't you want to buy more of our mice to make your favorite antibody, right? I think that's a big part of this.
They're almost human, but not in the legal and ethical sense. So.
Yeah, do with it as you will. But actually, I do want to say that I think that this is a big trend. I'm not sure if we've spoken to this directly in some of our journal discussions, but there's this idea that this technology has always existed, and now that there's this huge global pandemic, it's a great way to like showcase what your technology can do, right? Like I think that that's a really big thing about the Moderna vaccine. It's like, they had some candidates, they were developing some, but now they're going to showcase it and be like, right, like, this great new platform we have, like, don't you want to use it, like, and to sort of gain popularity. And I think that this paper falls into that camp for sure. Showcasing a technology.
I think that that's why these coronavirus papers can actually be quite fascinating. So I understand that not entirely pure motives, but
still, well, I mean, like, basically, like, if there wasn't a pandemic, this paper still would have come out just with a different viral infection.
Yeah, and it would have been
right like that.
It would have like made as many waves at all. I mean,
exactly.
The three frontrunners are all like next generation vaccine candidates, like the ones that everyone talks about, but no one's talking about the heat-killed viruses, even though those ones are the ones that like tried and tested.
Like absolutely, and the sort of the manufacturing methodology is like super well known, right?
Yeah.
And like maybe it can be scaled up better. Like I think that's another element. Or is like
Yeah.
where is the scaling going to come from in all of
I
this?
mean, all the infrastructure is already in place for those. So I mean, I mean,
Hmm.
it's amazing that those ones are the. I mean, yeah, I probably shouldn't be. We can moan about this a bit more next week, I think, on our off day.
Yeah, yeah, that's true. That's true. Yeah, let's see what we got next.
Yeah.
What's next on the
Oh,
figure
we've got some
docket.
neutralization assays.
Nice.
So
Of their top nine
yeah,
candidates. So from all of those pools, they've characterized that, these mice make human-like antibody pools. So now they don't tell us, right? That we don't know where they're from.
no, we have no
So
idea where they're from.
yeah.
So
This is the gap in the paper, right?
yeah,
They're not connecting those two things. Because maybe they don't need to. That's not the focus of this paper. They want to sell us on some efficacy of these antibodies now.
maybe they all came from the human candidates.
Yeah. Wouldn't that be a shame?
That'd be such a shame. All of them came from the human candidates. Okay, well, just not mention that. We're just like.
Yeah, and neutralizing. So this is our first step. This is where we're seeing, like, so much of what we're seeing now in the phase 2 trials, right? This is the output, right? There's nothing more than you can do but show neutralization. I think they're using pseudotype particles in this. They do both, maybe.
Yes, they do both. I think this one was with pseudotype particles. I can't. Let me check.
Yeah. Well, in the table S1, they do say pseudotype particle neutralization. So if people recall, that's not the SARS-CoV-2 virus. That is the VSV virus,
Yep.
some other virus. But they've replaced some of the surface proteins with the spike protein of SARS-CoV-2. So neutralization here is blocking the ability of that spike protein to bind to the receptor ACE2.
Yeah. So you want to have the lowest antibody concentration for the highest neutralization. So, like, if your curve curves to the left in that graph, it's good.
Yes. Yeah. The faster it comes up, right? The faster the earlier it comes up.
Yeah. Left is good. Far right is bad. So that's
Yeah, and they have a sort of a negative control,
Yeah.
right? Where they have it's an antibody against ACE2, I
yeah,
believe.
ACE2s. Well, this is actually quite kind of interesting because it's not really a negative control. It's actually like they actually mentioned like, oh, other competitors are using an antibody like this. They didn't say they didn't use the word competitors. They go, other
A
groups
subtle jab.
are using this antibody. It's like kind of the Mr. Clean advert where look at rival brands are terrible, but our brand is better. So
For sure.
So yeah,
And I mean, actually, that's a very strange therapeutic. I wouldn't understand why you would want to block, because like also ACE2 has other effects. Why would you want to add an antibody against that? It's a little bit of a straw man, right? Of a negative. Of a negative to choose.
But I mean, the thing about with like COVID outbreak is that people are so desperate that they will resort to something like that. And I think I have seen something like that because it kind of makes sense. Like, oh, we don't know spike protein, but we can block ACE too.
Absolutely. But they do have a real negative control. They have just IgG, IG1 isotype. So that's just the random antibody.
Yeah.
Bulk antibody. Mm-hmm.
Okay, so next is a big old graph that.
Next, it's like this is the SPR data.
Oh yes.
So
service plasma resonance.
Service plasma resonance. Yeah. So we had ELISA. That's one way to measure, right, antibody binding. But there's also this device. It's actually, when you see it, it's a big box, and then you get little cartridges, and then you load up your antibody into the cartridge, and then you test it on the big box. And it's a device that measures binding through a very strange physical property called surface plasma resonance, SPR. And the way I think about it is just that if something binds, basically your antibody of choice is flowed right through a flow cell, and in that flow cell is a piece of gold that's lined with the ligand. So in this case, they're using the receptor binding domain, right? And so that's stuck on here, and as the antibodies bind to it, it sort of changes the shape of the surface, the piece of gold, and then that is a readout. You get a readout of it.
I mean, the actual readout is, I tried actually a long time. I spent quite a long time trying to understand it because then I realized it's quantum physics, and so this.
Yeah, it's like a funny property of gold.
Yeah, I mean, all metals that are shiny have these things called like they have this sea of electrons, which is the reason why they reflect off things. So when light goes on, like hits the surface, it excites some electrons, and then they bounce off. But like depending on the type of electrons and the state that they're in, sometimes they might absorb stuff, and that can be manipulated by depending on whether like an antibody is attached to them or not. And that's basically the level of understanding I have. Like, okay, right, if you breathe on the metal surface the wrong way, it'll absorb slightly differently because these particles are plasmons aren't really particles; they're like kind of just effects of electrons interacting with each other. And then I went cross-eyed and I couldn't understand any further. But
I think but that's a good point. Like it's super sensitive, right? Is that idea? And actually, I think I misspoke. It's not the antigen that gets bound to the surface. It's the antibody. Yes. So it's like their antibodies are bound to that piece of gold, and they flow over the antigen. So they flow the receptor binding domain. And so yeah, as those antibodies pick up receptor binding domain, that very sensitive quantum effect gets read out. And you get a binding domain. Well, I guess what's nice is you can control temperature and stuff, right? Like ELISAs are generally done under like a really similar set of circumstances, but this one you can you can measure like when things fall off, right? Because like you can flow the receptor binding domain, and then you can flow no receptor binding domain, right? And you can see how quickly do those things disengage. And so basically you get like these very precise numbers of like how sticky the antibody is, the affinity of the antibody against the antigen. So that's all those K constant values. Like if anyone remembers chemistry, like the equilibrium constants, that's what this is. It's a way of measuring like the equilibrium constants of binding for antibodies.
I mean the advantage is that it's also label free, so the readout comes directly from that binding and unbinding, so I don't have to wait for an enzyme that's attached to it to have a fluoresce or have a reaction. So I think that that's at least that's what I saw from the BioCore YouTube video that was trying to explain this.
But you know, at the end of the day, what we learn is these antibodies are good.
Yes,
they are binding to the receptor binding domain at high affinities.
Yes.
Okay, so what else do we got here?
We got more neutralizing assays.
But now we're changing it up, right?
Yeah.
We're doing some cell. Oh, they've made this. They're using different cell types. To do these.
So in B they use PVSV in human like lung cancer cells, and again showing that binding and
the pseudotype virus.
And SARS-CoV-2 they use that again, but in viral cells. And so now it's not ELISAs. We actually have like cell death basically is the readout of this. Well, I think for VSV it might be, I think it's a GFP VSV, right? So I don't think it's cell death in the VSV case, but in the SARS-CoV-2 they're looking at cell death as the readout. I couldn't really figure out what the difference was between C and D, apart from the fact that they look at different antibody concentrations. So D actually looks at a much wider, like more within that treatment, like they actually have a lot more kind of resolution on the actual point where it neutralizes, whereas
they zoomed in a little bit.
They
zoomed out a little bit.
They zoomed out a little bit because it seems like they tested a bit too high antibodies. So for like the ones that could bind at very low rate, that could bind very well, they didn't get all of their
they couldn't see the differences as well between them.
It could be they didn't expect to see it like that, and they want to let us know they didn't expect that, so they can go look at deal and be like, oh, look how good our antibody is. It's
surprised on how good it was.
Yes. The next
but this is nice. This is very, I mean, I think at the beginning of this, like people were struggling with these assays, right? Like we were seeing like sometimes they wouldn't even have pseudotype or like actual viral infection. So like, we're well into this pandemic and the research methods associated with it, and we're seeing the data as we'd like to, as good as it gets in cell culture.
And next one, they look at the furin cleavage site. So testing whether that has an effect.
This is very interesting.
Yeah. So
because it's born out of observation, right? That that perhaps there's a difference in the infectivity of viruses with this furin cleavage site mutation.
Yeah. So they test the wild type cleavage site, then do this monobasic cleavage type, which is basically what's seen in the original recipe SARS-CoV-1, and then they knock it out completely.
Yeah.
And they find a difference in like the human cells, but not in the viral cells. And I believe we have touched on this before in our TMPRSS2 like paper that we looked at a while back. And I think they found like some kind of very similar things because I think.
Yeah, that not all the cell cultures behave the same way.
Yeah.
But that's also because of what it's because of the proteases they have, right? That do the pre-processing.
They have multiple different proteases, and some of them will cut no matter what, and some of them it varies. But But it's interesting.
Right. And I guess that's the other important thing to remember. Like all of this is like we're just looking at the readout of neutralization, but from that TMPR space, it's like happening in the lysosome, right? Or it's happening in the endocytosed particle bags.
The replication organelle.
Replication organelle. Whatever they are. Pre-replication organelles, probably.
Yeah.
They're like some molecular hybrid between a vesicle or a lysosome and a replication organelle.
I mean, the important thing, it does have an effect in human cells, whereas like viral cells, they're from a green monkey, so they aren't going to be as relevant as the lung cells. So that's next.
Yeah, they're able to have it neutralize.
Okay, the next one is let me see where this.
Oh, they want to see if just the Fabs S4 is it just the binding part, or do you need the full length antibody?
Yeah, that's right. And
yeah.
And
actually here, I actually find this kind of interesting. I didn't really know what to expect from seeing data like this. Maybe that's my naivete about antibodies being worked. But they see that the Fabs work worse.
Yeah. So, I mean, because I did get this idea in my head that all you need to do is block spike, and then you've solved SARS coronavirus. But actually, there's all these other things that antibody does that are very important.
But in this assay, it's just like it's just pseudoparticle neutralization. So, like, you know what I mean? Like, there's no other elements inside of this. I guess there is a cell culture at the end of the day, so there could be other stuff. Yeah. So like there's, I guess what I'm saying is the constant region in an antibody, usually we think of that as like an immune cell touches it, right? And then that causes the effect. But here, there are no other cells in this mix besides the cells being tested. I guess it's possible that there could be complexes and stuff that form on the FC.
I think it might be the the complexes, the chloromes. So what they say is like it pinches one and then it pinches the other and causes like the virus to clump up, perhaps, or causes something
like serum mediated effects.
Yeah,
because these assays are happening in serum.
Yeah,
so it could be serum effects.
Yeah, I mean, does that mean that they get complement? Because if they have complement, and then that could be another.
Yeah, I think complement. Well, in cell culture they usually deplete complement, right?
Yeah,
because the complement can mess up with things.
Yeah,
but I mean, there could be other proteins that don't get depleted because it's like a crude way that you deplete complement, right? Like heat treatment of the serum. So like there could be other stuff maybe.
But I think the main idea is like the clustering and clumping. I think is the thing that they say would be the big thing.
Yeah, that just FC regions associating with each other in some way.
Yeah.
Yeah, I mean, I think that this is kind of, it's interesting, like interesting information, but I'm like, I don't know what's happening in the humans. It's not really helping. It might be like helping like form like a hypothesis, right?
Yeah.
Like what's the most important thing inside humans? Well, they keep going, right? They do way more of these functional tests of the antibodies.
Yeah, they do. So they also look at neutralization.
So S5, S5. Antibody-dependent cellular cytotoxicity.
Yes. Let's see if I can pull up S5. Yes.
So for cellular cytotoxicity, this is So if the antibody recognizes something, will a cell like disgorge cytotoxic components? Will it kill the cell?
Yeah.
Kind of deal.
So what they do is they take these these cells and they get them to express spike, and then they add in NK cells along with the antibody to see whether that has a difference in causing them to drop dead. And so they have like blood from all three donors, and I think serum from and then they also like look at control versus spike.
So this could be kind of seen as like NK cells. Those are innate immune cells in this case, right? That just surveil and kill stuff. That, I guess the idea is that they would kill things that are sort of covered in these antibodies, right? Like, because that FC region again has these immune functions that like it activates immune cells. So here it's like cells that get covered in antibodies get killed by NK cells. It's like proof that the FC region is working, that it's the human region is working. This would be one of those things where I would really like to know which were the human antibodies and which were the ones that came from the mouse. Yeah. Right. Because like this is where it's like coming into play. This is like almost further characterization of how good the human antibodies actually are at doing things that are sort of human-like, I guess. Yeah. So it would be nice to know, but they don't show us that. Yeah, because to me that like in terms of like the comparison between the models, these are kind of like the comparisons model information that you would like to know, right? Are those FC regions working the right way? But I guess maybe they're the same. They're genetically identical, so maybe that's why they don't really show it, because it is exactly the same as it was. I don't know. Proteins get made differently in different bodies. Yeah, there's that potential.
I mean, I feel it would strengthen the model if they could say, "Oh, these did come from the mouse," but I mean, we don't know that.
Yeah. I guess the other thing to say about this is that this could also be like thought of as a substitute for CD8 killing. Yeah. Right? Because like NK cell, it's like a similar idea, right? Like when cells kill other cells, natural killers or CD8 positive T cells, activated CD8 positive T cells. So it kind of is trying to cover both those ideas. Yeah. I think it's cool that they get them from people.
Yeah, that is quite cool. Yeah, and the next like figure is basically a table showing the same thing.
Yes. Yeah. Well, I guess what's important here is that these Jurkat cells, right? They're prepping. Is it that they're prepping the cells? Because Jurkats those are a cell line.
Yeah, those are a T cell lymphoma line. And they basically they took some controls and they also implanted them with spike protein. I don't know why they chose a T cell line of all, but maybe that's because I'm not familiar with them.
It's because it's all about the assay setup, right. I think, right? It's bringing these cells together creates the effect, right? Because inside of the body, you have all the cells, they're working together to kill things. In this version, they have to sort of artificially recreate that. So that's also why that the Jurkat cells got transfected with CD20. It's like they've artificially made like active T cells, right? These T cells are just active. Yeah. And when they have that SARS-CoV-2 spike protein, then they have the antigen that gives them the specificity. What else do we say here? I want to look at the figure legend. I feel like there should be something more to describe.
So,
this is table S3.
So yeah, I mean, at this point they are starting to pick out like front runners for their favorite antibody, because like you've got three that come up quite a lot, which is Regen-10989, Regen-10987, and Regen-10933, and like those are like the three like hero like antibody. And they do talk about 10934, but they will that one is not doing less well. So
yeah, that's true. These assays are certainly helping to find the better antibodies that are coming out from this whole process. Yeah, let's move on. Let's look at the next one because it's like more of the same. Now it's a different type of assay. Now it's phagocytosis. So instead of killing the cells that have right, it's at different. These are basically targeting all the different ways the antibodies could work. Right, we don't really know how antibodies would work in the human body. So in this in vitro study, they covered their bases, recreating all the potential mechanisms for an antibody to work. And so they did their receptor binding. That's like the ELISA style. They did killing of the cells that are expressing spike protein. That's sort of the cytotoxic path. And then this is maybe the macrophages and things are eating the viruses before they even get into there. So that's what we're looking at here, phagocytosis.
Yeah, and you can see that again, we've got our three front runners like up at the top, the 989, 987, 933, and at the bottom is 10934. So we already see that there are winners and there are losers. 10934 not doing so well.
And I believe the difference between A and B, these are our controls, I think, right? So, that in A, they're not making the spike protein or something like that. Yeah. And figure S3. Oh, sorry, S6, right? Is that where we're at?
Figure S6. Let's see. This is S6.
Yeah, so like because you the cells have to be making the spike to be eaten up in this. Yeah.
And then we've got like some more numbers to basically add to that.
The table is just like the table that goes along with these. Right, the EC5, that's like the midpoint inflection point basically on the graph. I think is what it's measuring. That's very similar to like when we see ELISAs. I think they always give us like the 50, the binding constant in the middle.
Yeah. So basically, if you want numbers, this is what will give you those delicious numbers we need for when we actually have to plan experiments for real.
Yeah. And again, the takeaway from all of these is that in the ways that we think antibodies work, these are working. Yeah. Okay, let's see what's next.
So next they do deuterium. So let's see, epitope analysis using this fancy new technology involving deuterium.
I wouldn't call it new though. I don't think it's new.
It's probably not new. It was new to me, but then anything that's like newer than 10 years ago is new to me. So I'm So I'm just constantly like happy about everything. Where did I put the?
I mean, that's just like the pace of technology, right? We're seeing like so many more things. I remember hearing about this back in grad school, but I didn't think it was that widespread use. The way they make it sound, it's like a machine that does it for them.
Yeah.
They just like load up the machine with the plate and then this technique is applied. I don't think that was, at least I don't remember that being the case with this type of mapping before.
Yeah, so it's interesting because like, what they do is they take because all proteins have hydrogen that can bind and unbind to it, and
because everything's in water, right?
Yeah, everything's in water.
That's a big.
Yeah, exactly. Hydrogen like bonding is a really big thing for proteins. So and there's
everything's covered.
Yeah, everything's covered. So what they do is and deuterium is basically heavy hydrogen. It's hydrogen but with an extra like neutron on it. So basically it makes things heavier. So if you coat, so what they do is they basically dip.
It's you've heard that term before, heavy water.
Yeah,
right?
heavy water. So yeah, they take their proteins and they dip it in the heavy water effectively and create this, so that they effectively get a coating of heavy water. So it's almost like I mean I think of it as like being deep fried slightly. And whatever has the crust on it is exposed on the surface. But obviously, if you crack it open and see that meaty insides, you see that hasn't got the crust. And so that's kind of how they figure out where it's binding. Because when things are bound, there is no room for that deuterium to go in. So because of that, they can put through a mass spectrometer and look at all the things that are slightly heavier and figure out that those are on the surface. And the bits that would usually be on the surface but aren't heavier are the bits where they're being bound.
Yes. So they do that in a big panel.
Yeah.
Right. Because they want to see. What is if they bind to the same spots. If they can, if there is a set of antibodies they can use that bind to different spots, I guess, is their interest.
Yeah. So they really want to do this antibody cocktail. So they want to have antibodies that bind to different spots because they're worried about mutational escape. So a virus can mutate in one area and potentially render antibody treatment like unviable. But if you have two antibodies, that makes it a lot less able to escape, because then you have to have two 1 in a million chances happening, and that's going to make it a lot more stable. So that's why they've been searching for.
Yeah. I'll say though that that's not the only way, right? Like that's not the only way that you can make sure that an antibody, there's no escape from an antibody, because the other thing is that what if the mutation to escape breaks the function?
Yeah.
Right. So like they're also like, this is not the only way to sort of go about solving this problem. But it's certainly, because they have such a wealth of antibodies, it's a good approach to take.
Yeah. And I guess they're trying to make sure that they don't bind in the same place with it, or they don't interfere with each other if they're used.
Yeah. They want to find the ones that don't overlap, I think, so that they can create this cocktail.
Yeah, and it seems like a lot of them are like, I mean, the red pairings don't work. The yellow pairings, and yellow is like, white is like almost like no.
What? No, white is good, right, for them? Yeah, white is good for them. And you can see their setup here, right, is that, and it's kind of like the deep frying thing, because they're just dipping things, right? Yeah. They like dip. They have, they capture the antibody that they're interested in onto like a plug, I guess, and then they can switch out the things that they dip it into to try to catch the RBD that's labeled. Yeah. I think, right, the RBD is labeled in some way.
Yeah, I think, and they check for that, the competition.
I think this isn't related to the the deuterium assay, right?
I think they basically take those assays and they structured it and picked out the areas of the structures that overlap. So if if the binding footprint overlaps a lot, then they say it's bidirectional competition, and then that.
Oh no! I think actually it's different. No, this is just a separate assay to look at the competition.
Oh, right.
And then it's the next figure that then they investigate using that method, the deep fry method, to actually identify the surface.
Yes.
Yeah. Yeah. So actually, this method here. It's just again what binds to this RBD that they labeled. They labeled some receptor binding domain, and so if the receptor binding domain doesn't stick to the antibody, they say, oh, it's already been bound by the other antibody that's sitting in that well. And then so that's what they're saying here. But from that, they get a sense of like which ones don't compete, and then they take those unique ones and then they say, okay, well, let's actually know something about, in some ways, confirm where they don't compete. And that's what we're seeing in figure S7.
Yeah.
Is the results of now the deuterium thing, where I guess black is no data. In green, right?
Yeah.
Things that have negative delta. Is that a double negative?
I think that these basically, like, there's a diagram of the receptor binding domain of the spike protein and showing the areas that the antibody's bound. So purple, it means that's got a high affinity, and black has got a low affinity. So they're looking at not only like how much affinity they have, but also how much crossover. So
right.
So I've got a slide where I'm just focusing in on like the top two winners, which of
that's nice to see.
So we've got like REGN-CoV-2 which has it's basically on the left side. There's a lot of binding on like the green side of things, and then REGN-CoV-2 is on the right side. So they tend to be looking, so it kind of confirms that they're both on different sides of things. And I mean, it's interesting because I mean 09087 is, I mean, in the previous competition assays, that was the one that basically is binding in a very unique place compared to all the other ones in. So that's quite interesting. And 93.
Yeah, they're diving down now, right? Like after identifying that there's some antibodies that don't compete in a binding assay, then you want to dive down to that mechanism and say, okay, well, that means they probably bind to different places.
Yeah, I mean, going back, so they basically class the binding into, I think, 4 different families based on the areas that they bind, and from that they looked at the. So I mean, because the antibody that was doing really well is 989, which in EC2 is like this, got these big purple patches, and it would be so if you're just using one antibody, that would look really good because it binds quite heftily on both sides. But because it binds like slightly in the green side, that means that it will interfere with the other antibodies. So that's why they can't use that in that specific cocktail.
Yeah, I guess I didn't even see. Did they comment? Does this like I realize now part of the confusion is because this is figure 4,
yes.
right, of the graph, not figure S7. And then figure S, did they talk about it? Because we should see the same patterns come out, right? Like in figure S7 to figure 4. I believe it does.
Yeah, I think they do come out very, very similarly. That's why I thought that they were the same data.
Right. And they're gonna not end there. They're gonna bring it to the highest standard, basically, of structural biology and give us a structure.
Yeah.
Through our favorite method of cryo-electron microscopy. Single particle, though, so in this case, they mix them together and then they take pictures of just the mixed bound sort of complexes. So not whole cells here. They don't have to blast away things with ion beams. But the same idea that if you see things multiple times from different angles, you kind of get a sense of what the whole thing looks like through shadows. And that technology has come a huge way. The table beside here is just showing us some of the parameters that you would normally get. I think what's interesting to remember here is that, right, like the measurement of this is they have all this disparate data. It's all just like pictures of the molecules in different positions, and that all has to come together in a model. A computer has to construct it, and actually that's like a full-time job, right? Like people do this as their living. They like take all this data, algorithms help them, but then it also has to match to like a reality of the protein, right? Like the protein we know is made up of different amino acids, and like they have to fit together. So that's what some of these things are, right? Like when you see like the validation and the Ramachandran plot and the RMS deviation, just to say that it all fits. It is all like the protein that you think. This is the protein that you think it is.
And they show that they can bind, the two of them can both bind a spike protein at the same time, which I think is pretty special and pretty interesting. And
absolutely. Can bind the receptor binding domain of the spike protein.
Right. So we don't know exactly what does the real spike.
It's like pretty good evidence, but you know, in the biological context, it's still a little bit out there because like there's a huge protein that receptor binding domain is attached to. It doesn't have the same degrees of freedom necessarily to move around as it does in this circumstance. But yeah, pretty good. And these are fabs too, right? That they're using. This is just the fab domains that they're adding here. So I don't know. Like this, if you shouldn't come away with this imagining that like this cocktail in the body is like giant antibodies are like all converging onto the same virus. It's possible, but I don't know if that, you know, we don't know if that's happening.
I can fantasize about whatever I like, and if that's what I want to believe in my head, that's what I will believe.
But it's kind of interesting. I never even thought about. To me, I don't even think that much about like multiple antibodies binding to something. Like when I think about polyclonal antibodies, you know, not just a single clonal antibody, I think of it more as like covering all the bases, right? Like, oh, like just in case like binding here like isn't as effective, like you bind this area and it's like it works. It's funny to be presented with like this intense structural data saying that it can definitely bind two at once. But I mean, good from the perspective of, I mean, you know what I'll say? It's definitely a strong argument for like mutational, like, you know, protecting from the double mutation or whatever, because it says that like in order for a spike to escape, it would have to have this whole surface changed. And that bodes well for, because like that might mean functional change to the RBD.
Yeah.
And so if it means functional change, then like that's as good as, right, never happening.
Yeah.
Because yeah, if there was functional change, then yeah, you just wouldn't have an infection anymore.
Yeah, I think that's all the figures. And so I guess it brings us.
Yeah, so it works. It works in vitro.
Yeah.
And I did want to say, though, I sort of mentioned this earlier, monoclonals, we use them in other therapeutic circumstances. A lot of immunomodulatory drugs are monoclonals, and they're expensive. Because as you saw in the workflow that they had here, they make them in cell culture. They make them in Chinese hamster ovary cells. And if anyone's done cell culture research, you know that like growing cells, yeah, you can grow a lot, but it's like on these like thin plates, monoculture. Of course, there's technology that helps people scale that up, but it is difficult to keep cells in like large cultures like that.
Yeah, if you look at this scale, they need to have like giant vats of cells, and that's an entirely different level of science. That I mean, we're bench scientists. We never had to deal with like those kind of bioreactors.
Yeah, it does exist. It does exist. Not to say that that tech isn't there, right? Like it's just that it's like a very complicated thing. And if you think about scaling up like bacteria versus scaling up, you know, something like this, like there are significant challenges.
Yeah.
But people love it because I think this customizability of antibodies is like, that's why people love monoclonals.
Oh yeah.
And yeah.
I mean, they are very expensive at the moment. I mean, well, I think you're quoting me prices.
Oh yeah. Yeah, I was saying that like so Humira is like this like is like the most prescribed monoclonal antibody out there, and like a single or maybe 2 doses of that costs $5,000, and like I don't know how much is in there, and how long would someone have to be on therapy for an infectious disease, right, to see that effect? Yeah. So you know, there's lots of questions to throw out there, but this is not a drug that is going to be cheap when it comes. Not a therapeutic that will be cheap when it comes to market.
Yeah.
But what is really nice is that you know, unlike some of the, you know, what is nice is that this like is if you have an active infection, you could really use this. Right? Like if you had high viral load, this is the time to use it, because like these antibodies are for the virus.
Yeah.
So yeah, it's good from that sort of critical care perspective. I would love to see like some of this style of therapeutic get out there, because I do think it would have some use in saving lives. Yeah. Maybe not everyone's life, but some people's lives.
I feel like kind of the same argument, because theoretically this is basically the same as convalescent serum, except it's manufactured and it.
Yes, so theoretically absolutely.
It should. Except you don't have to rely on having to get it out of patients or having to rely on like blood supplies. You need to it's a bit more scalable.
And it's more controlled too, right? Like you also don't have to worry about some of the side effects of being given a cocktail of antibodies from another person.
Yeah.
Right? They've picked out what they think are the important components and made it in vitro for us.
Yeah, and I feel like if they do build out the infrastructure for this, for baking large amounts of antibodies, I mean, even when this outbreak's over, they can still use that for lots of other things, because it's not specific to coronavirus, though, these techniques.
Yeah, absolutely. I mean, that's kind of like a whole other thing, an area of research, right? Like just like all of, if this actually ends up being a therapeutic, that's going to put huge amounts of money then into making the therapeutic, and that's going to be research in a totally different realm that we've never touched on right, during this series. But that research that goes into how do you grow Chinese hamster ovary cells to 10,000 liters, 100,000 liters. That stuff is, well, I think a lot of that research is super proprietary, but there are engineering journals, and some of those lessons trickle out into the greater world.
Yeah, I mean, so we need 8 grams for, we're getting 50,000 cases per day, so 8 times 50,000
Yeah, that's what you're saying. You're saying the president received 8 grams of this.
Yes, that's what I've heard. I mean, I could be wrong. I mean, I'm not saying that exaggeration is something that Trump doesn't do, but it's.
Yeah. Well, I mean, in this case too, it would matter, right? Like it does matter. Dosing, it's a big deal because it's expensive to dose people more and more of this.
Yeah.
Yeah.
So yeah, that'd be I think 400,000 grams. So that's a lot of antibody, and that could cost a pretty penny. And
yeah,
and of course, like the question is like exactly what stage what stage of infection do we give it? Is it best early, late? I think it might be good at all times, but
yeah.
I mean, there are a few like studies out in hamsters and in primates on bioRxiv that only just discovered after picking this paper, but I still stand by this because this is where it was created. So, I mean, I mean, based on this paper, you don't know exactly what it'll do in an actual infection, but I mean, that's something we will learn in the future. This is more about how it was made, which is interesting itself from science, because from our perspective, that's the interesting part, not the
I was gonna say, like,
is it dead yet?
Yeah, I guess it would be nice. Maybe we could have briefly viewed like the actual efficacy because it is out there in animals. But yeah, in terms of talking about like the development process, I found this far more interesting because like we know in animals, it's like, oh, they're gonna slice up the lungs and they're gonna show us the pathology and the better pathology.
Treatment research is great, but a lot of it is it dead yet? Why isn't it dead? What's happening? I mean, it gets a bit morbid, so. And we still got our vaccine. We still got like some vaccine stuff in this. So they treated a mouse to actually an entirely new, because we've seen like kind of
just the spike protein boost.
Yeah.
Instead of that's interesting.
So like that'd be an interesting idea, because if that's successful, then perhaps like partnering up the Moderna and the Novavax could work, because Moderna's.
I mean, once there's some approved vaccines out there. There might be, if the pandemic continues to be a problem after that, there might be studies that are done where like these types of different strategies of vaccination are tried out with approved vaccines.
Yeah,
I could see something like that. Yeah, well, that's what we got for you today. If that's interesting to you and you want to join us to select the papers that we normally deep dive into, that's what we'll be doing next week. You can tweet us any of your paper suggestions or questions that you see with the hashtag or directly at us. And that's all for now.
Yeah. Thank you so much for joining me. This has been great. I will see you next week. We're doing our off show. We'll be looking at a bunch of different papers. If you have any suggestions, obviously you can tweet us or look at with the hashtag, and we'll try to discuss your suggestions.
Mm-hmm.
So thank you. Bye.